Yes, we are continuing the binary fission notes and hopefully getting into our goodness gracious, cell cycle regulation notes. So the plan is to get that done today or get as far as we can today. On a Monday, we are, goodness gracious, where am I? Monday, we are going to talk about what happens when we don't follow the cell cycle or the cell cycle can't be regulated. We will review all of this stuff. Wednesday or Thursday, we will do progress questions on AP Classroom. Then we will go over them, and if we have time, we'll do something fun. So, cool, 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 cool. All right. Essentially, we're just finishing off the semester like we would finish off any other unit, because time kind of snuck up on me. Um, and we don't have finals, so that's plus. So we are going to continue binary fission and then hopefully get into cell cycle regulation. All right, let's see if I remember where we left off. Two origins of replication. I think so. Yes, beautiful. Okay. So, the entire process of binary fission. Step 1, replication begins. I'm going to have our little, that's our bacteria cell. We are going to have, let's see, so we are in a circle, so where it starts is where it has to end, and we are going to have a little origin of replication. But our origin of replication has already started to divide. So our DNA replication begins, I guess is what I could. Yeah, DNA replication begins. So our little origin has started to split off. We are going to have one side of this DNA replicate over here as we go through our chromosome. So that is step 1. Step 2 is replication continues and the cell elongates. Replication continues and cell elongates. So we'll draw a little bit wider. At this point, our origins are towards opposite ends of the cell. Origins of replication at opposite ends of the cell. So replication at this stage isn't complete. So somewhere in all of that tangle, there is still one circular chromosome being copied into two. Step 3, replication is finished. Replication finished. And plasma membrane is pinched, and if applicable, new cell wall is deposited. So this is something a little like that. And those are connected. My board's a little off. So I still have the origins of replication highlighted because if when these become their own cells and when they go to divide, that origin of replication is going to be the same. So where this cell on their chromosomes started to replicate, these cells will also replicate. And then finally, step 4, we have 2 new daughter cells. This process is a lot simpler than our mitosis cytokinesis process that we learned about with eukaryotes. So this happens instead of mitosis. We don't have to worry about nuclear envelopes. We don't have to worry about multiple chromosomes. We just duplicate the DNA. As it duplicates, the origins are pushed to opposite ends of the cell. DNA separates and gets pinched off in the middle. Doing okay? Any questions so far? All right. So that's a binary fission. I'm going to briefly go through our mitosis evolutionary connection. Because it is important to draw connections between past and future content. And the whole point of biology or of AP biology is to get you to draw connections and think critically about this content. We are going to make as many evolutionary connections as possible. So, first thing we need to know is prokaryotes preceded eukaryotes by over 1 billion years, which means prokaryotes were on Earth almost 1 billion years before eukaryotes came around. Prokaryotes preceded eukaryotes by over 1 billion years. So it is hypothesized that mitosis evolved from prokaryotic mechanisms. So, here are some some pieces of evidence that support this hypothesis and why it is still just a hypothesis. So, evidence. The biggest piece of evidence that they have at the moment is that some proteins involved in binary fission are very, very similar to proteins involved in mitosis. So, proteins involved in binary fission are closely related to proteins in mitosis. They have similar functions, they have similar structures. They're not quite the same, so there's probably some changes over time because of the changes in what the process needed. But that's our biggest piece of evidence, is that we can look at the proteins involved in binary fission and we can look at the proteins involved in mitosis and see some very glaring similarities. Let's see. There are also variations in the process of cell division. So even within mitosis between organisms, there are slight variations. Even in binary fission, there are some processes that are a little bit more complicated. And there are some processes that are a lot simpler. And we can kind of use those as steps to connect mitosis to common ancestors. So variation in cell division exists. And it can be used like steps to connect mitosis to common ancestors. Can be used to connect mitosis to common ancestors. So there's our evidence. But there is also some issues with our hypothesis, and that is the fact that we can't observe the process of mitosis in extinct species. So all of the steps in between, all of the common ancestors, we can like trace lineage back based on cell structures and such because we have fossils, but we can't observe mitosis in those cells. It's not like we have all of the stages of cell division fossilized. So what we do have is existing species as examples, but we have to ignore any intermediate mechanisms because we don't have physical evidence. So here's our problems. Can't observe cell division in extinct species. So we can only use existing species. And can't rely on any proposed or potential intermediates. So while we have evidence to support this hypothesis, there are also strong arguments against it, or at least some holes in logic. Not because of lack of research, but from lack of evidence. Because when something's extinct, we can't really observe the process of cell division. So, any questions about this stuff? Comments, concerns, complaints? Mm-hmm. We can propose connections, but we can't draw any distinct conclusions because we don't have the ability to watch the extinct one replicate. So we have to rely on other bits of evidence to prove connections between living and extinct. We'll talk all about that when we get to evolution. All right. So that's all I have for you for binary fission. We are going to kind of trek our way at least a little bit into cell regulation. I want to give you the time to do the exit ticket that was supposed to be for yesterday, but we didn't get to. So this one's not too long. We might have to like do the graphy bit on Monday, but c'est la vie. Cell regulation, or cell cycle regulation, more like. We know what a cell is. We know how a cell divides. We don't know how a cell knows to divide. We talked about the different phases in the cell cycle, the G1, S, G2, M, and then all of the phases of M. But we haven't talked about the cell cycle control system. Cell cycle control system. There is a cyclically operating set of molecules. So there are molecules within the cell that their concentrations rise and fall in a repeating pattern. And those rises and falls of concentration trigger and coordinate the events of the cell cycle. So everything that controls the cell cycle relies on these molecules. Cyclically operating set of molecules that trigger and coordinate key events in the cell cycle. There's a kind of a really good metaphor for this, and that is thinking of the cell cycle as a like washing machine or a cycle in a washer. If you look at the like little dial, if you have one that like moves with the with what is happening inside the washer, new washers just have like mechanized stuff, but the older ones have like dials that move as the washer spins through the cycle. So it will go on its own, but it responds to, or sorry, it goes on its own based on a preset timer, but it's regulated by internal and external checkpoints, meaning at some point in time, we have to control it with actually pushing the buttons. So cells can respond to external signals, but they also respond to internal signals. So there's a sensor that tells the washer when it's filled up all the way. And there's a sensor that tells the washer that all of the water has gone out of it to move to the next part of the cycle. So let's see if I can get that into cleaner words. So, a cell cycle proceeds on its own according to a built-in clock. But it is regulated by internal and external controls. And it also has checkpoints. The washer is not going to enter the spin cycle if there is still water in it. So just as the washer stops and waits for all of the water to get out, the cell will stop the cycle and wait for the previous step to complete. It's a little messy. Okay, so these checkpoints are really important, especially when it comes to loss of control, which we'll talk about Monday. But these checkpoints are points in the cell cycle where we need stop and go ahead signals. Control point, where stop and go signals regulate. And there are three really important ones. There is one towards the end of G1. We have to finish G1 before we can start synthesizing DNA. There is one at the end of G2. Mitosis is not going to start unless we've got everything we need for it. And G2 is all about prepping for mitosis. And there's one in the M phase. Mitosis requires a heck of a lot of cell signals because we need to make sure that cell is definitely ready to do its dividing. So, I talked about an internal clock, right? That clock is rising and falling concentrations of two molecules called cyclin and cyclin-dependent kinases. The cell clock is made of cyclins and cyclin-dependent kinases. We've talked about kinases a lot. They are very important to the cell's functioning. Yes, fluctuations in these two regulatory molecules control the cell cycle. Fluctuations in the concentrations of these two regulatory molecules control the cell cycle. If we take a cell that is not in division, if it's in some sort of like there's another phase in the cell cycle called G0. That's just a cell that's not dividing, it's just kind of dormant. If we took one of those cells, injected it with cyclins, it would automatically start dividing. Automatically. No prep, just mitosis. So, let's see. Yeah, I'll give you the definitions of these two molecules and how they work, and then I'll let you do the exit ticket. Yeah, so these protein kinases, you guys have talked a lot about protein kinases. Pretty important because we're going to write that definition again. A protein kinase is an enzyme that will activate or inactivate other proteins by phosphorylating them. Protein kinases is an enzyme or are enzymes that activate or inactivate proteins by phosphorylating them. There are special protein kinases called cyclin-dependent kinases. Cyclin-dependent kinases are only active when bound to a cyclin molecule. Only active when bound to a cyclin molecule. Because they have an active and inactive form, these cyclin-dependent kinases, or CDKs, are always present in the cell. It is the cyclin concentration that depends on activity. Actually, I might have time for this. So, the activity of cyclin-dependent kinases increases or decreases with the concentration of cyclin. Activity of our cyclin-dependent kinases increase or decrease with changes in the concentration. Ran out of space there. Of cyclin. If you are a fan of shorthand, I have this written in my notes as essentially hieroglyphics because my notes say CDK activity rises and falls with the changing concentration of cyclin. If you're a shorthand person. Oh yeah, we have time for this. We're gonna draw a graph. So there is a special CDK complex. I'm gonna change colors. There is a CDK complex or cyclin-dependent kinase called MPF. This stands for maturation promoting factor. Or some people call it the M phase promoting factor. So if we were to look at a cell or look at a bunch of cells, we've got a graph where this is time, and we've got it divided up into M phase, G1, S, G2, M, G1, S, G2, M, G1, etc. All right, we're just writing the cell cycle. And this is going to be our concentration. We're going to start with our MPF, right? So our MPF is at its highest concentration when the cell is in M phase because it promotes mitosis. So it'll look like this. There's none in that G1 and S phase, but it rises and falls like that. Then we have our cyclin. So our maturation promoting factor is a cyclin-dependent kinase. So its activity depends on the concentration of cyclin. So if we're graphing the concentration of cyclin, we are going to gradually rise until we hit peak M phase, and then it will fall. And then towards the end of G1, it will start to increase, start to increase, start to increase, hit mitosis, and fall. It will stay at a low level until we hit the end of G1, rise again, rise again, rise again, and fall, and so on and so forth. So if we were to take a bunch of dividing cells and measure the concentration of these cyclin-dependent, or the activity, rather, sorry, of the cyclin-dependent kinases and the concentration of cyclin, it would look like this. So from this, we can definitely tell that the activity of mitosis, whatever triggers mitosis, is dependent on the concentration of cyclin molecules. Questions, comments, concerns? Beautiful. All right, there's an exit ticket on Canvas. If you could fill that out for me, please. And then Monday, we will talk about checkpoints and what happens when cells ignore those checkpoints.